Hi, good morning, everyone. Welcome to Lunch with a Scientist. I'm Leah with Headwaters Science Institute, and today we're going to meet Kelly Loria from the University of Nevada, Reno. She's a wonderful scientist that's going to talk to us about mountain lakes and streams. But before we get into that, just a quick reminder that our summer programs are available for registration now. Sign up now because we think they're going to fill up fast. So they're designed for high school students to explore research through hands-on curiosity about anything in the world that they're interested in. So the students will pair with a professional scientist mentor, go through the process of developing a research project, creating a research question, doing data collection, analysis, and then even creating a formal research presentation and authoring a research paper, which we will publish in the Headwaters Research Journal and also have students submit for formal publication in a youth journal. So it's a really cool beginning career experience for a high schooler, whether they have science experience or not, or are interested in a science career, or maybe just a little bit curious. A little bit of something there for everyone. There's a female-specific section, the Girls Summer Research Experience, and then there's a section for everyone called the Headwaters Research Experience. And so you can check out both of those programs by going to headwaterscienceinstitute.org/science camp. Please sign up. Like I said, I think we're going to have them fill up. And you can also access financial assistance applications there on our website. So with that said, I'm super excited to introduce Kelly Loria to you. She's a PhD candidate in the Natural Resources and Environmental Science program at the University of Nevada, Reno. She graduated from the University of Colorado Boulder with a degree in ecology and evolutionary biology. And her background is in alpine lake ecology with some experience post-graduation having led a team monitoring the alpine lakes in the Green Lakes Valley of Colorado. She's broadly interested in aquatic ecology, especially in headwater catchments, which we'll learn more about what those are today. And her more recent research interests include investigating patterns of metabolism and nutrient uptake by algal communities of mountain streams in the Lake Tahoe Basin. So very pleased to welcome Kelly Loria.
Hi, thanks for having me.
Thank you very much for being here. We're super excited. And before we jump in, I'm just curious, as most people know, we're based here in Tahoe and you're down in Reno, not too much change in elevation there. We got a little snow last night. How about you?
Yeah, we did too. And hopefully we might even get a little bit more this afternoon. So it was a cold morning and a nice light snow, though. I think it pretty much either melted or sublimated outside.
Mm-hmm. Well, of course, as our watchers may or may not know, snow is very important to us in California because what happens here in the mountains becomes our snowmelt, which then feeds our watershed for the rest of the state. So I'm curious, in studying this watershed when it's snowing, are you still out there doing field research?
Some of our research continues. We have a few projects that are looking at the salinity concentrations in the streams around Reno. So those are up and going and have sensors in them that take real-time data throughout the day. Some of my work, because it depends on water temperature as a factor, gets a little bit thrown off when it gets quite this cold. But yeah, for the most part, we're out there regardless of the weather. And I'm personally very grateful for the snow. I love recreating in it. And yeah, it does help provide a lot more water going into summer. So if we could just get a little bit more snow to fall this year, it would be really great.
Mm-hmm. Well, we've got a week of snow forecast, so hopefully that'll feed your water research, and we're super excited to hear more about it. So I'll let you take it away.
Awesome. Thank you so much for that introduction. Yes, I am Kelly Loria, and today I will be talking about mountain stream ecosystems and nitrogen. So to start off this talk and give you a little bit more information about myself, as Leah mentioned, I'm coming from Colorado, and this is that lake that I have experienced studying. It is one of a few alpine lakes that are part of a long-term ecological research program, or an LTR network. There are about 28 of these sites spattered throughout the US, so they're a really great organization to get involved with too for in-college research. And this one in particular has a very long data record dating back to the 1940s. So as an undergrad and as a postgraduate student, I helped maintain part of that record. And in that maintenance, part of it included this instrumented buoy, much like the streams that I'll get into later, where there are different water quality sensors deployed at different points along this line. And it can get really pretty bogged down with algae, as you can see in this photo. The other part of the lake ecosystem that's really important are these two microscopic plankton species. And at high elevations, they actually grow quite large. So even though these photos were taken on a microscope, as you can see, that scale bar is in the millimeters, they're large enough to see with your naked eye. This is just like a quick clip of them floating around in a jar that's hopefully working okay for everyone at home. And so, because alpine systems are pretty much free of human contact directly, all of the change we see in these systems tends to occur indirectly because of things that humans do down valley, and then those consequences get transported into the alpine systems. I'm going to get into the details of some of those processes in just the next few slides, but some of the things we've been seeing we are able to test. And so these large tubs in a field are testing some of the different dynamics as like miniature lakes in a field. Great. So one of the examples of indirect impacts of human activity on climate change and mountain ecosystems is the fact that we use combustion for travel, industry, as well as parts of our agriculture. All generate these types of greenhouse gases. So some of these are NOx and SOx, which you may have heard about in your classes, which are either nitrogen or sulfur-based particles, as well as carbon dioxide from cars and traffic. But we also get these sources of methane and ammonium from agriculture, as well as oil drilling. So all of these particles are emitted at somewhat of a concentrated area, typically at the lower elevations, and then they get transported upwards into the atmosphere where they serve as a condensation nuclei, or just a thing for water molecules to adhere to when clouds form. Then these clouds actually are able to travel really large distances in the atmosphere. If you think like things that are emitted over Sacramento, say, can freely travel until they start to hit the foothills where they experience the topographic roughness of a foothill or a mountain. And that rough spot in the landscape helps those clouds to condense and start to get different types of orographic movement within them that leads to precipitation. So then those particles are deposited through this process known as wet deposition as ammonium, sulfate, and nitrogen in aquatic ecosystems and mountain ecosystems in general. So one of the central themes of my research is figuring out how capable mountain ecosystems are of tolerating this excess nutrient loading from human activity. Specifically, I'm interested in how mountain stream ecosystems and how mountain stream ecosystem function here as rates of gross primary productivity or ecosystem respiration change as a function of nutrient availability in these kind of iconic mountain streams that you can find throughout the Tahoe Basin. To orient you a little bit more to this idea, I'm just going to go over the components of a mountain stream ecosystem. And apologies if this is a little bit redundant for some of you, but some of the main sources of energy in this ecosystem, like all ecosystems, is the sun. And so plants, here depicted as this tree, this shrub that could be a manzanita, some aquatic macrophytes, these are algae, all are capable of doing photosynthesis, which is the process by which these plant organisms use light from the sun as well as water and carbon dioxide to create energy, and oxygen is created as a byproduct. So moving along this kind of imaginary food web, we have insect or macroinvertebrate. We also likely have those plankton that you would see in the lake photo, and at the higher trophic level, we have something like a fish, which could be, say, a Lahontan cutthroat trout, which is a species of particular concern here in the Tahoe area, maybe a sculpin though. So these larger trophic organisms are able to consume those primary producers and gain energy that way. But in order to break that energy down, just like all omnivores consuming organisms, they need to undergo respiration. So respiration is the process by which we use oxygen to access sugar and energy from our food, and carbon dioxide is created as a byproduct. Interestingly, plants also respire because they're only able to access that light source during the day. So at nighttime, when there is no sunlight available, these plants actually continue to utilize sugars that they built up throughout the day and release some carbon dioxide as a byproduct. Now, all of these sort of metabolic processes that are happening every day, every minute of every day in a stream ecosystem result in waste. Just like humans, all of these species produce excrement, they shed, and all of that organic matter starts to build up in the stream channels and undergo decomposition, which is another process that consumes oxygen and can produce carbon dioxide. So, moving on to that research question, why does it matter that some of these nutrients might be artificially transported into these fairly pristine stream ecosystems? Well, because these systems are so pristine, they tend to be somewhat limited in the amount of life that they can hold due to the available nutrients that just naturally persist within a system. So we consider these types of ecosystems to actually be nitrogen limited. So when an additional source of nitrogen can come into a system combined with some other micronutrients that just help kick off growth by providing subsidies for other different types of metabolic processes, algae, and mostly algae, can grow really rapidly and to a prolific amount. So these algae are typically single-celled plant organisms that reproduce on the rate of minutes to hours. And so they can rapidly take advantage of that nitrogen coming into the system and grow, but because they're short-lived, they're also dying at really high rates. So it starts to cause organic matter to build up in the systems as well, which can lead to an increase in respiration. All that respiration and decomposition could lead to poor water quality, which can become pretty unsuitable for some of these higher trophic organisms like that cutthroat trout, as the water starts to become saturated with CO2, which means there's less room in the water for oxygen. So it becomes harder and harder for that trout to process its own food and access to the dissolved oxygen that would be in the water without all that added growth. If this condition continues for a really long time, it could even make these type of stream ecosystems inhabitable for these higher trophic organisms. So just rooting that graphical demonstration in some actual real images from this fall and late summer in the Lake Tahoe Basin, you can see these ecosystems that should be fully flowing aquatic ecosystems actually start to stagnate, where we get pools starting to form, and actually water is just trickling through this part of the channel. So it's still moving, but at a really low rate. We also start to see these really prolific green mats form on rocks, as well as even some floating chunks of algae, which is interesting because some of these algal species are actually able to trap air bubbles within their large clonal mats, and they use that buoyancy to then lift up from their place on the stream bed and float downstream, colonizing a new area, which can lead to nearshore greening in Lake Tahoe, which is not really a favorable condition for recreation. It makes the rocks slippery, but it also means that the oxygen is getting depleted from the nearshore communities in Lake Tahoe, which is really harmful for some of those littoral or nearshore species of fish, as well as insects, which there's a number of native to the Lake Tahoe Basin. So to get a handle on this situation and how these streams are acting metabolically at this point in time, I have several streams with these instrumented with these dissolved oxygen sensors throughout the Lake Tahoe Basin on both the west and east sides. And these sensors take rapid measurements of dissolved oxygen, which I then use to estimate rates of ecosystem primary productivity as well as respiration, and then eventually water quality. So picking a stream to do this type of work in is not quite as straightforward as it seems. Out of each of these images from A, B, and C, which do you think would be the best type of system to go out and monitor? And mainly, what are the big differences between these pictures? Well, if you are thinking that this middle picture might be the most accessible stream for this type of research, you would be correct, because stream A is basically a waterfall. So it's undergoing a lot of aeration, which is this physical process where that cascading of water over rocks actually drives oxygen from the air into the water and hypersaturates it, which is great for water quality. It is not great for estimating how much dissolved oxygen is coming into the system just because of biotic contributions. And this other stream, C, is again a great stream, but it's kind of a wetland at this area of it. So it's actually got some stagnation, probably more organic matter that's trending it artificially towards a higher respiratory signal than would necessarily be true for the entire reach that I'm interested in analyzing, and not as generalizable to the entire watershed catchment. So one of the metrics that we need in order to get at these rates of primary productivity and respiration is actually going out and collecting stream flow. And stream flow is done by basically walking up to a stream. We lay a transect, which you can think of as like that continuous, if that yellow line was continuous across a horizontal section of the stream. And then we go and we collect these little point measurements along the transect of how fast the water is moving at that given point. As stream water speed actually changes as a function of stream depth, because the bed surface of a stream is an area of roughness. So it tends to be slower right at the bed and it tends to be slower right at the surface. So actually the fastest velocity in a stream tends to occur at the 6/10 depth. I know that's an improper fraction, but it is something that the USGS, which is one of the largest federal agencies that measures water, uses. So it's kind of a standard metric to describe here. So then we have this transect laid out and we go along with an acoustic Doppler, which is just basically a machine that has a propeller that counts the amount of rotations. So we use that as a metric of speed. And we take a metric of depth as well as a metric of width. And we imagine each of those little areas as a small little volume estimate, or like one of Q1, if you will. And then we go and we sum all those Q sections for one horizontal cross section of a channel and get an entire estimate of how fast that water is moving for a given point in a stream. Next, like I've said, we have these dissolved oxygen sensors, which we basically use a cinder block to keep a proper orientation in a stream channel. So that way there's less measurement noise and they're very stable in the location where they're being measured. And they measure oxygen by measuring the amount that can diffuse across a microscopic membrane. And they're basically just taking measurements every 5 minutes of the dissolved oxygen concentration at that point in time, as well as the water temperature at that point in time. So we need to use an additional metric from weather stations to be able to actually get at what the oxygen saturation potential can be in these streams. So we aggregate weather from these type of weather stations. And I'm including this picture here of a SNOTEL station, because if you are ever hiking in the Tahoe Basin area, you might actually come across one. Typically they're enclosed in a fence to discourage people from coming up and like getting a little too curious about the instrumentation, but they have all of these really useful metrics within them that tell us really interesting things. So right now, SNOTEL stations across the Tahoe Basin are using the snow pillow, which is just this undisturbed bed of snow that actually measures the full weight of snow that's accumulated. So it's really important not to like step on those or the snow above those if you see them at ski resorts and stuff, because it will actually compact the snow and make it a lot harder to measure. And then they actually use optical lasers to measure snow depth, snow temperature, as well as they collect these metrics for the amount of sunlight coming in and solar radiation, wind speed. What I'm most interested in is air temperature as well as barometric pressure. So next, we aggregate all this data we've either collected in the field or downloaded from nearby weather stations, and we use it to estimate these rates of gross primary productivity, GPP, ecosystem respiration, or gas exchange, K. So some of these metrics, such as those observations of water temperature, stream flow, as well as barometric pressure, are needed to estimate the rate at which gas is exchanging in and out of the water column. So you can kind of think of that K as like a metric of how much of a waterfall are things acting, like how much gas exchange would naturally be occurring in this system based off the turbulence of the water that's coming through it at a given time. Next, we use this equation developed by Odom in the mid-60s to get at how much primary productivity and ecosystem respiration might be occurring in one of our streams. And some of the parts of this equation I've pulled out for you just to understand a little bit better. We use an estimate of the final outcome of this equation that we actually kind of use to rearrange to algebraically solve for gross primary productivity and respiration is the rate of change in dissolved oxygen for any unit time. And then we have Z here, and which is the mean breach depth of a stream, K, that gas exchange coefficient, as well as the dissolved oxygen that our sensor observed in the field, as well as the dissolved oxygen that is actually saturated potential. That you can use one of these old school graphs that has like a 3-dimensional space in it to estimate what saturation potential would be. But honestly, we have a modern computer algorithm that I just run my data through and calculates it for you. So I won't labor that point. Similarly, this is its own series of math equations to derive that use all these components, which is why it's highlighted in this black box here. So next, I'm just going to share what some of this sensor output data looks like with you all. So this is a time series plot that starts on September 28th and runs through October 26th and has various qualities of dissolved oxygen that the sensor goes ahead and downloads, and then I transform for that algorithm for saturation potential at both a given time as well as overall saturation. So this top plot is just the raw concentration of dissolved oxygen from the sensor. What I'd really like to point out is how variable this data is. So as you can remember, when the sun is out, photosynthesis is taking place. And so you get these peaks in oxygen where biologic communities in the stream are actively photosynthesizing and giving off all this great oxygen as a byproduct. But in the nighttime, you get these troughs where the sun's down and those organisms are still alive. And so they're still consuming sugars within their body and therefore contributing CO2 into the system. So we see these troughs. What's kind of funky about this is this data set extends into the fall, which means that water temperature got a little bit cold here, and the amount of oxygen that water can hold is somewhat dependent on its temperature. So it's a physical deterministic relationship that's called gas solubility. And so what's great about correcting for oxygen saturation is you can see it flattens out that curve, so you don't start to get like a false linear pattern within your data. And then we can see the oxygen concentration per any given unit time of one of these observations. Next, we use that to calculate these metrics of gross primary productivity in a package that the USGS puts out called Stream Metabolizer in R. And what I'd like to just note is that gross primary productivity is positive, which is great, and it's also variable over time. So it's kind of a little bit of an interesting rate that seems fairly dynamic. And there are standard error bars included around these metrics. So it shows like the degree of confidence that I have that that value likely occurred at a given point in time. And it's a rate, so it's measured over in this, so it's gross primary productivity per day. So it is measured over time. And ecosystem respiration can actually be negative and somewhat mirrors the pattern that you would expect from gross primary productivity, as they're somewhat inverse processes. So getting back to that central question, how does mountain stream ecosystem function? How are that green line and that brown line related to nutrient availability in these mountain streams throughout the Tahoe Basin? So what we do is we actually go out and we collect water samples. We typically use this little syringe just to pull water directly from the stream and then push it through a filter. And then we have our water samples that we process on a various amounts of analyzing machines back at the laboratory at University of Reno, Nevada. And this photo is just to show some of the struggle of that wintertime fieldwork. This is actually a stream gauge location that we sample from in Colorado, but it can get buried in like up to 3 meters of snow. So we do this work year round, and it can be a little bit trickier in the winter. So we use these nutrient samples to get an estimate of the amount of nutrient that is in the stream for a given sample, but then we also estimate the amount of nutrient that has likely been deposited throughout the system and subtract the value in our sample from the amount that likely is coming in through deposition and standardize it for stream flow or the water at a given time. So that way we have just a standardized concentration for the flow rate. And we assume this concentration for a standardized area within a stream that we're measuring. So here are the results of that analysis for 2 different types of nitrogen species, a total nitrogen as well as a nitrate. And this is gross primary productivity and ecosystem respiration against nitrogen load for 3 different streams, Blackwood Creek, General Creek, and Ward Creek, all on the west side of Lake Tahoe. And what you can see is there doesn't appear to be a strong relationship at this point in time with this preliminary data between gross primary productivity and the nitrogen load per given time. Interestingly, though, we do start to see this slight negative trend for ecosystem respiration and nitrogen load, but the trend is most pronounced at Ward Creek and a little bit more muted at both General and Blackwood. So future work is to basically improve upon this data set, both through added techniques statistically, but also with a larger series of streams that we're analyzing and a greater resolution of time. So statistically, we intend to take this deterministic or mechanistic model for how we measure and estimate these rates and parameterize it through a statistical model to see how those lines on the past plot could be better trended to fit the data that we're seeing. And then we can even bring in external sources of data from past sampling efforts in these systems through a machine learning technique to start to get at what patterns are we really seeing and how likely are those patterns to be occurring given the data that we have. So this technique is called Bayesian statistics, and it uses prior information to better fit. It uses prior information to better fit what our expectations for our future results could be, and then we're able to actually get legitimate descriptions of our model outcomes and make really strong statistical statements that are something along the lines of, I am 89% sure that this magnitude of gross primary productivity is occurring for within a given unit area of these streams, which is a little bit stronger than some of the classical statistics you might be learning in class right now, which have to do a little bit more about the idea of infinite samples and repeated tests. And then we're able to actually use those strong statistical statements to make ecological predictions into the future, and actually tell managers of these watersheds what our findings are in the hopes that some of these strategies could be used to reduce that nearshore greening and keep Tahoe blue. So in summary, humans can have really strong impacts on mountain ecosystems, even when they're doing things very far away from these systems via atmospheric transportation and deposition. And these impacts can really influence the amount of algae growth or gross primary productivity happening in the streams, as well as the amount of CO2 saturating the water and ecosystem respiration, and therefore just the quality, the aesthetics, and the function of these systems. And sensor technology and these large time series data sets is helping us actually get at these problems and create a little bit more effective management strategies around some solutions to keep these ecosystems pristine and in the best quality for recreating. So lastly, I'd just like to give a brief thanks to my advisor, Joanna Blaszczak, as well as the rest of our lab members, my committee members, Aaron Hayden, Dr. Adrian Harpel, Sudeep Chandra, as well as the Whittle Forest Wildlife Area, Tahoe National Forest, and the USGS for all of the contributions they make and just having all that data and land access available. And with that, I'm happy to take any questions. Thank you so much for listening.
Thank you, Kelly. That was great.
Great.
So this is especially cool for me because Ward Creek is sort of in my backyard. So tons of questions for you to jump in.
Lovely.
Yeah. I wondered, so when we're talking about a stream or waterway that has very little ecosystem respiration happening, what's likely the cause? Is it always too much presence of algae, or could it be kind of lessened stream flow from objects blocking, or yeah.
I think, that is definitely part that I'm hoping to get into this summer a little bit more actively with a wider range of stream expression or phenology, where, in the start of the season, you have these really strong snowmelt pulse and you have water rapidly moving through, and it's likely that you won't have much production happening because scour is just occurring at such a great rate that biologic communities don't really have a chance to grow, and also the nutrients that are coming through the systems that help them grow is like just being flushed downstream really quickly. So as the season progresses and you start to get those more discontinuous pools forming in channels depending on the water year, so in dry years you would expect more of those pools, a little bit more stagnation, not as much mixing, a lot of organic matter building up, and probably really high rates of ecosystem respiration, and maybe more like hot pockets of ecosystem respiration for a given stream. So just the stream might become less coherent, and it actually might get much more decoupled from general metabolic patterns because it will just have a lot of different types of habitat occurring within it.
And so I also wondered, as we're talking about the way waterways can change, do you see impacts in ecosystem respiration if there's erosion or too much moisture that change the
Great
waterways so that
question.
you don't have streams, instead you have little pools?
Yeah, I think erosion is a huge issue, and I can understand where you're coming from in Ward Creek. It's definitely got some really incised channels and a lot
Mhm.
of exposed dirt. And dirt is actually pretty full of nutrients. So yeah, it can be a little bit of a mixed bag depending on what like the disturbance, if we're gonna like generally just call it that, could be. Something like erosion where you could have these new nutrients coming into a system. If it happens in the spring, it probably won't have too much of an effect for the stream. It might have a bigger effect on the lake because it will probably just move through the stream quickly. If it happens in the fall, it could have a pretty crazy effect on some pools in the stream and really kick off respiration. But that being said, I think it does kind of depend on when and where, which is a horribly vague answer to give you. So apologies. But yeah, I think it is a metric that's very sensitive. But most of this work has pretty much this type of modeling work has really only been done in lowland systems, and there's a few other groups that have done it in mountain systems. But yeah, it's kind of a novel thing to be approaching. So I don't have the greatest handle on how exactly certain disturbances will shift the balance between whether the stream is primary producing, absorbing carbon dioxide, or really highly respiring and maybe even emitting carbon dioxide. But it's typically seen that most things that disturb regular stream ecosystem function will be bad for rates of primary productivity and CO2 absorption, and likely create a system that is respiring more and potentially even emitting CO2 to the atmosphere.
Hmm. So I know you've had this wonderful fortune of looking at a lot of very rural lakes, which are fairly pristine. And to move to the other side, I think there's always a concern about human impact. And we've seen really massive algal blooms in the Great Lakes. I'm
Yeah.
wondering, how are algal populations typically impacted by human presence in a waterway? And what can humans do to stop the spread of them?
Oof, yeah, that's a big question. Also, it is a little bit species dependent because algae are like a pretty broad group of organisms that can come in all different shapes and sizes, and some of them are better adapted to certain conditions, such as like warm water or higher nutrients. And actually, some of them like there's a certain group called diatoms, which make these really beautiful glass frustules or cases, and they're like cold water specialists that tend to be indicative of really pristine, great environments. Those fluffy mats of algae that tend to bubble up with air and be easily transported downstream tend to be unwanted nuisance species and sometimes even harmful species. And some of those, such as cyanobacteria, are actually capable of synthesizing their own nitrogen within themselves. So they're able to handle a wide range of ecological conditions. And it's potentially theorized that temperature and warm, they might be warm tolerant species. So I would say it's likely still a mix of most systems in the mountains are nitrogen limited, but a lot of systems in lowland ecosystems are phosphorus limited. So whether there's a source of nitrogen or phosphorus coming in, like the Chesapeake Bay classic example is mostly a phosphorus case too, because it's where all of that agricultural runoff hits a saltwater environment and it just kicks off rapid phosphorus transformations that then make that nutrient more accessible to all that algae, and then rapid growth, rapid death, and kind of not so great water quality ensues. So it is highly variable across ecosystem type, which is a really interesting part of this research for people to get involved, because and asking this question in almost any body of water that is nearby you and you care about is still a really, really good thing to do with your time and interesting to get results on.
Sure. So do we need to be thinking about whether we encounter algae if we're accidentally somehow bringing that on our clothes or on
Oh yeah, I would
equipment to other areas?
I completely agree. So, and I think if there are any fly fishermen out there, you might recall that in a lot of places, felt waders are banned because certain types of algae cells can maintain on the bottom of a fabric surface and they can withstand drying. And there's actually some really kind of horrific case studies in New Zealand where we brought this, America brought this species of algae called didymo or rock snot that is like this really fluffy, really slippery algae that can handle very cold, clean water and just forms this like clonal mat that completely inhibits a stream and changes the entire community of organisms that can persist in that stream because it just blankets the bottom of it. Yes, that as well as chytrid fungus, which is really common in the Sierras and impacts frogs really negatively. Yeah, a lot of things can be spread from soil. So it's always, yeah, always a best practice to try to clean off your boating equipment, your water equipment, and even really your hiking equipment if you're being very safe.
Mm. And then, so just flashing back to chytrid, we had a presentation specifically on chytrid on frogs, I think
Nice.
2 or 3 weeks ago. So if that piques your interest, please feel free to look back. Always connected.
Excellent. Yeah, because that's one of those other like high mountain things that's wild. You don't expect these mountain systems where they're hard for people to get to. So you think they'll always be pristine and withstand humans and our impact. But yeah, they're not above it all, even though they're very high up.
Mm. So I'm wondering if you could break down gross primary productivity a little bit simpler for
Yeah,
us. I know that it's a metric that you come up with by looking at a lot of
totally.
things. I'm wondering, as a scientist, have you gotten to a point where you can kind of see a waterway and kind of anticipate what the gross primary productivity will be, and what are some of the indicators of that?
Maybe one day. I'm sure there are some senior scientists in the field that might have that magic power. I am way too early in my career to have a confidence in that sort of estimate. But yeah, it's basically the amount of carbon that an organism is synthesizing into biomass. So kind of the
Mhm.
bulk that these algae and plants are putting on in general. So any organism that can do photosynthesis is a part of the gross primary productivity estimation. So any sort of thing that has chlorophyll and can synthesize its own energy is included in that estimate. And mostly that's plants, to be really simple. There's some weird stuff in microbiology where organisms can be a little bit, they call it mixotrophic, where they can consume and also produce their own energy, but it's very tiny things that you find in drops of water.
Mm. Interesting. Well, we'll give it 20 years and we'll have it back, and one day you'll be
Sounds
able
great.
to wave the magic wand. So, getting a little deeper into the work specifically that you do, you didn't talk too much about it in this presentation today, but you let me know beforehand that you really do a lot of math,
Yeah.
and that math wasn't always your favorite subject, but
Oh, no.
it's part of the research that you're doing. Can you tell me a little bit about your journey with math, and what does the math side of your work look like?
Totally. Yeah, it is. So a lot of it, I showed a few of like the physical or deterministic equations that basically I have not created at this point in my career, but other scientists have gone out and measured these relationships in all sorts of different conditions and decided that based off these physical constants in the environment, you know, it's most likely if you were to draw a line between these things that you're measuring, that is what the shape of the line would be in the terms of an equation. So I think really math clicks for me when it's thinking about the shape of things. So math is really just representing these physical shapes that we have out in the real world. And those shapes can be really dynamic and change. So having an open mind for some of the ideas about where things that you might think like learning probability theory from a deck of cards is really tedious and boring, but we use probability theory to get at an idea of how likely this relationship is based off the data that we have in hand to observe. So it does all kind of scaled up if you're interested in pursuing a scientific career. That being said, I'm like constantly having to reread exponent and power laws for things that I'm trying to figure out and solve. So it's not something that you always need to have completely memorized and dialed, but it is helpful to feel comfortable accessing it. And I think things like natural science give you different opportunities that are just like outside of maybe some of the really classical like statistics and algebra examples and calculus examples of like if a train's coming to a station, to start thinking about things that you care about and getting ownership over like I want to know what that rate is and that speed or that shape or the likelihood of that. So I do think for me, it helped when it all kind of got shifted into a vocabulary of things that I cared about, which are just the environment.
Mhm. Interesting. Very interesting journey that you've incorporated something you didn't love into something you do love, and now they're sort of married together.
Yes.
That's great.
And I think it is nice because yeah, it's really cool to coding too. It can seem a little bit scary when you're not super used to coding, but it's pretty gratifying to have things instantaneously change. So sometimes getting to work out some of this math in like a more computer coding language is a little bit more friendly and forgiving than having to do it all pen and paper. And knowing that you have the whole internet to research things to help you figure it out when you get stuck is really great.
Mm. So I'd like to wrap up with some inspiration for the students who are watching. So a couple questions. If someone's watching and they sort of know that they want to follow the educational path that you did and end up studying rivers and streams and stream ecology, what educational path would you recommend for them?
Oh yeah. I think it definitely college is a big step in that journey. And I think being an advocate for yourself in your own education and realizing that you might not be good at everything that you need to get to get your foot in the door in a college university setting, but to really feel strong in the strengths that you are good at and pursue it because as a scientist honestly there's all these sorts of different skills. So we've talked a little bit about math, but writing is a huge component of it. So honestly most of my day is split between math or writing, and then there is some science that's just identifying organisms in a field, which is botanizing. And there's a wide variety of skills that make for successful scientific careers. So I think getting involved in a lab and trying to be a strong advocate when you get to a university, and even participating in programs like this now where you're just getting an idea of that, but showing up, emailing people, being somewhat persistent about like getting people to take a chance on you, which is probably harder now in this times of remote communication, but definitely worth doing even before you're at a university if that's your next step. But looking through to see what professors are associated with that university in an department that you're interested in, and see if they just have opportunities to either have a low level paid job or maybe just volunteer on a couple days in their group on certain field trips.
Hmm. And then what would you say for a student broadly interested in science who maybe knows they want to study nature and doesn't know specifically what they want to do in the
Oh
future?
yeah, totally. Yeah, I mean, I think just go out and try it really, because it's not a big deal if you do something for a few weeks of a semester and you hate it. Sometimes that's like the best way to find out that you think that studying water is great, and then the tedious amount of bottle washing we have to do to get our season. We do so much dishwashing. um, that might be a make or break thing for some people. And so I do feel like, yeah, just getting out there and figuring out if it is a good fit is the best that you can. Also, they're, great resources. This one's probably a great resource to get a flavor for what different people's research looks like. But I think most scientists are really willing to email back and forth with people. So feeling okay in asking questions directly to scientists or their grad students or, professionals in the field is a really powerful skill to start building.
That's great. Thank you so much for the advice for our students and for presenting your work. Kelly, Loria, thank you for being here. I hope you have a
Yeah,
great
of
day.
course. Thanks, you too.